Hello students, today we are going to start the next biomolecule. In the earlier videos we have completed the introduction and then we have completed the first biomolecule, that is protein. We have also seen the amino acid and that amino acid, how it forms protein. Everything we have seen. Now after that we'll see the next biomolecule. So the next biomolecule here we are going to see is carbohydrate. Okay, so let us see the carbohydrate. So first thing about the carbohydrates is, what is carbohydrate? It is the polyhydroxy aldehyde or ketone. Okay, so this thing you have to know. This is the definition of the carbohydrates, that it is going to be polyhydroxy, means OH group will be present more than one, and it will be a long chain where we can have the end that can be either aldehyde or it can have the ketone group, right? So this carbohydrate basically are the sugars, okay, or this is the component of sugar. Okay, sugar will be simply here we will be called as saccharide. Okay, so it will be a saccharide. So whenever we see the carbohydrate, what are the components will be present in the carbohydrate? The elements are carbon, hydrogen and oxygen. What we have seen in the earlier amino acid, in the amino acid we have seen carbon, hydrogen, nitrogen, oxygen, and along with that we can have sulfur, we can have any another alcohol group, different type of acidic basic amino acid we have seen. So number of components can be present in amino acid. But in case of carbohydrate, we are going to see only three elements, and that will be carbon, hydrogen and oxygen. So the general formula of the carbohydrate will be CnH2O whole N. Okay, so it will be the general formula of the carbohydrate. Okay, what we will see in the next, that this carbohydrates, okay, this carbohydrate will be the sugar of your body that is required for the energy production, our ATP, that will be required, that will be produced in our body for the energy, isn't? So it will all get by the sugar. In our body we have the glucose, right? So glucose is a source of energy for our body, so that will be also a type of carbohydrate. Our stored food, animal stored food is glycogen, so glycogen is also a type of carbohydrate. So if you want to classify the carbohydrates, so we can get three types of carbohydrates. What are they? We can have monosaccharide. Okay, saccharide means sugar. Then we will have oligosaccharide. And third we have polysaccharide. Okay. So monosaccharide means in that sugar we will have only one sugar. So this is the simplest carbohydrate. So it is going to have only one sugar. Whenever it comes to oligosaccharide, oligosaccharide will have 2 to 9 sugar, okay, and 10 or more than 10, okay, that will be the polysaccharide. Okay, so we will classify, we will see each of the carbohydrates one by one. First of all, under the monosaccharide, how many type of sugar we can have, right? Then after that we will see a group of this monosaccharide that will be called as derived monosaccharide. Okay, so what will be the derived monosaccharide? These are the types of monosaccharide, but here only some changes occur, so we will see what are they. Okay, so under the monosaccharide we will see the derived monosaccharide also. Okay, so that will be the introduction of carbohydrate. So let's just move to the first, that is monosaccharide. Okay, so here we will see how many types of monosaccharide we can have. Okay, so first of all, if it comes to the monosaccharide, what will be the general formula? Okay, So see here, this is monosaccharide, which is going to have only one sugar in it. Glucose is an example of monosaccharide. So first thing, here the carbon, okay, will start from 3 carbon atom up to 7 carbon atom. Okay, that will be the monosaccharide. Okay, so because it is going to have a hydroxyl group, so see, more than one hydroxyl group should be present, as I already have told you in the definition of carbohydrate, that it is a polyhydroxy carbon. Polyhydroxy, it can be an aldehyde or ketone. So obviously it will have more carbon, okay, where the hydroxyl group can be added. Okay, so it is made up of 3 carbon atom up to 7 carbon atoms. So we can have triose sugar, okay, then tetrose sugar, then after that we have pentose sugar, then after that we have hexose sugar, okay, then after that we have heptose sugar. Okay, so what is the general formula of all? General formula, but sometimes it can vary, that we have seen CnH2O whole n, right? So if this carbon is 3, then what will be the number of hydrogen? It will be 6. Then once again O3. Okay, so this is the triose sugar. Next one, tetrose sugar, C4H8O4. Pentose sugar, C5H10O5. Right? Next one, hexose sugar. Already we have seen it earlier, C6H12O6, and heptose sugar, C7H14O7. Okay, so these are the 5 type of monosaccharide we can see. So from here, what are the sugars you need to know their structures? Okay, so we need to know the structure of pentose sugar and the hexose sugar. Rest of the sugars, the structure, basically the chemical structures are not important. You don't have to see this triose, tetrose and heptose sugar. You don't need to know. Under the pentose sugar, I have already told you in the earlier page that it is the monosaccharide, which can be also another type of monosaccharide. We can see that is the derived monosaccharide, right? So what are derived monosaccharides? See, suppose one example is a pentose sugar, one example is ribose sugar. Okay, so in this ribose sugar, what we can see, C5H10O5, right? If you remove one oxygen from it, okay, then it will be deoxyribose sugar. So it will have C5H10O4. So if it is this, then it will be a derived monosaccharide. Okay, so this will be a derived monosaccharide. So what is this example of this pentose sugar? In this pentose sugar, C5H10O5, that is the pentose sugar, that is called as ribose sugar, and this derived monosaccharide of this ribose is deoxyribose sugar. Okay, so ribose sugar is a chief component of RNA, the genetic material RNA, and deoxyribose sugar, it is the chief component of the genetic material DNA in higher grade of organisms. Okay, next one, hexose sugar, one example. This is very important, that can be glucose. Okay, it can be fructose, it can be galactose. Okay, so yes, these are the monosaccharides. After that, we will see the structure of monosaccharide. Before that, I need to tell you that a very important thing, normally the carbohydrates, not only the monosaccharides, are they optically active? Yes, they are optically active compounds. So yes, they can be either dextro or levorotatory compounds. Okay, so yes, they are optically active. They can be either D or they can be L. Okay, that is dextro-rotatory or levorotatory. So here these are the monosaccharides. They are generally D whenever we see the monosaccharides, we can see most of the monosaccharides, okay, most of the carbohydrates, okay, they are D. But yes, we have one example of N monosaccharide here, that is fructose. Okay, fructose is N or levorotatory compound. Okay, so this is about their one character, physical character, that they are geometrically, they are optically active molecule. They are either D or L type of, yes, we can see D glucose, we can see L glucose. So we will see what are the structures. First of all, in the monosaccharide itself. Okay, so under the monosaccharide, we will see the structure of the monosaccharide. So first of all, we'll see the structure of hexose sugar. Okay, what will be the structure of hexose sugar? Okay, one example is glucose. So you know the structure, chemical formula of the glucose, C6H12O6. Okay, so now in case of glucose, what we can see two form. Okay, it can be either chain structure or it can be ring structure compound. Either we can see the chain structure compound or the ring structured compound. So we can see first of all the same compound. So it is going to have 6 carbon, 1 2 3 4 5 6, right? So it is going to have this backbone. Okay, next one, okay, so here on this side we can have hydroxyl group. Here it is hydrogen, it is hydroxyl. Okay, so this is the structure of glucose molecule. So in this glucose molecule, you can see this is the first carbon, this is second, this is third, this is fourth, this is fifth, this is sixth carbon. Okay, so here this will be the third carbon. Okay, on the third carbon, what happens? The OH group will be shifting towards the left side. Otherwise, on the right side we will have the hydroxyl group, on the left side we will have hydrogen. Okay. Now this will be this chain structure. What will happen if it forms a ring? Okay, so just see, this oxygen will be shared by the first carbon as well as the fifth carbon. Then what type of structure we can see? That it is going to turn. Okay, it starts to have a develop a ring. So this structure will be the ring structure. So how this ring will be formed? See, a single oxygen will be shared by the first carbon as well as the fifth carbon. So I have told here 1 and 5. So this first and fifth carbon is going to share the same carbon, the same oxygen, right? So this is the first carbon, this is second, this is third, this is fourth, this is fifth, and this is sixth carbon. Okay, so that's why what we can see, that this is going to form a structure. That structure will be the ring structure. Then it will be a ring structure. So this ring structure, just see here, this we are going to put CH2OH here. Okay, here. Okay, so here we are going to put OH here. Here it's OH, OH, and here OH. Okay, so what is this structure? That will be the alpha glucose. Okay, so remember, this will be the alpha glucose. Yes, we can also see beta glucose too. So what will happen to the beta glucose? See, if in the first carbon, okay, if you see the first carbon and hydrogen is present above and hydroxyl group is present below, then it will be the alpha glucose. And how I have drawn this structure, you just see. First of all, here it is the fourth carbon. So first of all, fifth and sixth carbon we have seen. On the fourth carbon, you have to put H above, OH below. On the third carbon, you have to just change it above OH, below H. On the second carbon, above H, below OH. And on the first carbon, we have H above and below OH. That will be alpha glucose. Now, we have seen these two structures. One is strain structure and another is the ring structure. Yes, I will draw the next one, that is the another ring structure, but that will be the beta glucose. So what will happen to the beta glucose? See, there will be a difference. What is this? Here, third, fifth, CH2OH. Okay, here this is the same oxygen shared by the first as well as the fifth carbon. So here we have OH, then this structure will be called as the beta glucose. So what will be the difference between alpha and the beta glucose? C. So what happens in alpha and beta glucose? Here difference is this, the first carbon. Okay, so if it is on the first carbon, above we have OH and below H, then it will be the beta glucose. And if it is in case of ring structure alpha glucose, then above we have H and below OH. Okay, so that will be the differences between the alpha glucose and the beta glucose. So why these are important, I will tell you in the reducing sugars and the non-reducing sugars. So one thing you just see in this ring, okay, so in this ring structure we can see this ring is been formed by both alpha and the beta structure. Okay, this is very much important to know that this ring is formed by how many carbon? Fifth carbon. That means 5 carbon atom form the ring and 1 central oxygen. So this will be a ring structure. Though it is a hexose sugar, we have drawn the structure of glucose molecule. So in this glucose molecule, what we have seen, a ring structure which is formed by 5 carbon atom and 1 oxygen. So this type of ring will be called as the pyranose ring. Okay, remember this will be the pyranose ring. Now after that, the structure of glucose we have seen. Now after that, let's just move to the next structure, that will be the pentose sugar. So we have seen the structure of hexose sugar, both ring and chain structure, as well as alpha and beta glucose. Now after that, we are going to see the next structure, that is sugar, that is pentose sugar. An example of pentose sugar is ribose sugar. Okay, so on this ribose sugar, we will see both the chain as well as the ring structure. Okay, so just see in this pentose sugar, what we will see, the ring structure as well as the chain structure. So in this chain structures, just see, first of all, it is made up of fifth carbon, 3, 4, 5. Okay, so what will happen in this structure? So on the right hand side we will see the aldehyde, and on the left hand side we will have the hydrogen. So this will be the chain structure, right? So whenever we see the ring structure, what happens? Okay, so let us see the ring structure. Then this ring will be formed by 4 carbon and the fifth part, that will be our oxygen. That means this oxygen will be shared by the first carbon and the fourth carbon. Okay, so this structure will be like this. Okay, a single oxygen is shared by the first carbon and the fourth carbon. Okay, so let us see the structure. The fifth carbon will be attached to the fourth carbon as an arm. Okay, just as an arm it will be present. Okay, so the structure, how it will be like this. First of all, here this will be H, we'll put H. Next one, here it is H. Here we will have hydroxyl group. Next one, here we will have hydrogen. Here it will be hydroxyl group. And here on the first carbon we can see here we have hydroxyl and here it is hydrogen. Okay. So this structure will be a ring structure. So whenever we see a ring, so this is one example of the straight chain and the ring structure, and this ring formed by the ribose sugar will be called as the furanose ring. So this furanose ring is actually formed by 4 carbon and 1 oxygen. Okay, remember this will be the ring structure. But I have told you that in this ring structure, that is a pentose sugar, what we can see the derived type of monosaccharide also. So this derived type of monosaccharide, how it will look like, see, it will be the deoxyribose sugar. Okay, so deoxyribose sugar will have only this chains. That means on that second carbon, what we will have, we will remove the hydroxyl group, and hydroxyl group will be replaced by one hydrogen. So this structure, that on the second carbon, if hydroxyl is removed by the hydrogen, then it will be the deoxyribose sugar. Okay, so this will be deoxyribose sugar, and if this hydroxyl group is present, then it will be called as the ribose sugar, right? So remember this, these are the two structures you need to know, that is hexose and the pentose sugar. They are going to be very much important. After that, let us move to the next type, that are the oligosaccharides. So in the oligosaccharides, what we have seen. Okay, so first of all, here we have completed the monosaccharide. After that, let us see the oligosaccharide. So on the oligosaccharide, we can have how many sugars? We can have 2 to 9 sugars. 2 to 9 sugars are going to form the oligosaccharide. So here we are going to see the structures of the disaccharides. Okay, so rest of the sugars, that will be not that important. Disaccharides like maltose, lactose, these structures will be important. Okay, so let us see the structure of disaccharide. Okay, we will see the structure of one disaccharide, that is lactose. Okay, so let us see the structure of the lactose molecule. Okay, okay, so here what we will see, that first of all we'll draw the glucose molecule. Okay, so first of all, so in this structure, suppose I have drawn here, we will not draw this hydroxyl and hydrogen. So only on the first carbon, this is the first carbon, and here this is the fourth carbon, right? So in this lactose, what will happen, just see. Okay, so here we have the hydroxyl, here we have the hydroxyl group, here it is hydrogen, here it is hydrogen. Okay, so what will happen now, here the bond will be formed, that will be the glycosidic bond. So how this glycosidic bond will be formed? Here one water molecule will be removed. Okay, and whenever this water molecule is removed, what bond will be formed? That is glycosidic bond. Okay, so this bond will be, just see how this bond look like. Okay, so this bond will look like this here. Okay, so this type of bond will be formed. So this bond, what we can call, this will be called as the glycosidic bond. Okay, so here this glycosidic bond, what type of glycosidic bond it is? It is formed in between the first and the fourth carbon, right? So this bond is been formed, that is in between the first and the fourth, right? So as it is formed in between the first and the fourth carbon atom, so that's why this bond will be the 1,4 glycosidic bond. So this is the structure of lactose molecule. After that, let us move to the next another structure, that will be the maltose. Okay, so in this maltose molecule, what happens? See, there will be only one difference. What is the difference? Okay, so the difference is here we will have hydrogen. Here it is having the hydroxyl group. Here it is hydrogen. This is hydroxyl group. Simply, okay. So as this is the alpha structure, okay, alpha glucose. So what will happen here? This OH and OH, okay, that will repel. They will just water molecule will be removed, and as this water molecule is removed, one bond will be formed. That will be this. Okay, so this will be the structure of lactose. Okay, so this is the another type of disaccharide. We have already seen the structure of the lactose and the another structure, that is maltose, right? So after that, let's just move to the last disaccharide. So the next disaccharide is the sucrose. Sucrose is formed by one glucose and one fructose. Okay, so whenever you see the structure, see, this is a little different from the rest of the two earlier what we have seen, that is lactose and maltose. This sucrose is a little bit different from the other two disaccharides. Okay, what are they? First of all, here this is the glucose molecule. Okay, here we will have the next, that is fructose. Fructose is going to form a furanose ring, and here this is the glucose molecule which is having a hexose ring. So here this is CH2OH. Okay, so the first, this will be the first, okay, this is the second carbon. Okay, so here it is hydrogen, hydrogen, this is hydroxyl, hydroxyl group. Okay, now the bond formation will be taken, and here one water molecule will be removed, okay, and we'll have this glycosidic bond. So whenever this glycosidic bond is been formed, then it will be the structure of sucrose. Now a very interesting thing is happening here, that we have to know the reducing and the concept of non-reducing sugar. Okay, so what is the reducing and non-reducing sugars? As already what we have seen it, see, the normally the glycosidic bond, see here what happens, the glycosidic bond is formed. Normally what should happen, a glycosidic bond should be formed in between the aldehyde, okay, aldehyde group of one molecule, one monosaccharide, and ketose of the another monosaccharide, that will form a glycosidic bond. But what happens normally in case of this structure, okay, so in this reducing and non-reducing sugar, what happens, see, whenever you can see, this is suppose a structure of glucose molecule. Okay, so this glucose molecule here, this aldehyde is been free, okay, and it is forming a bond. Okay, so whenever it is forming a bond, suppose, then this is the next, here we have the bond. Okay, here bond formation is taken place. But what happens here in this structure of suppose lactose, what happened? One of our aldehyde group is free. Okay, the first carbon is free. So whenever the first carbon is free in this structure, then it can have the characteristics of the reducing sugar. Okay, but in case of non-reducing sugar, what happens? See, okay, in case of non-reducing sugar, what is happening? The first carbon here, okay, and this is the second carbon in this furanose ring. In the pyranose ring, the first carbon, and in the furanose ring, the second carbon. They are both are utilized in forming the glycosidic bond. So that's why if any of the carbon, first and second carbon, they have been already utilized to bond the form, there is no free aldehyde or ketone present. Okay, normally aldehyde and ketone are present on the top, the first end of the chain, it is present. Okay, now what is happening in this sucrose? Okay, remember in this sucrose, what happened? Already the first and second carbon is utilized to form the glycosidic bond, but rest of it, suppose on the disaccharide, what we have seen, lactose, okay, lactose and maltose. So in this lactose and maltose, though 1,4 glycosidic bond is been formed, then also the first carbon is free. Okay, in this first carbon we have this aldehyde group free, okay, of one of a ring. So that's why as aldehyde and ketone group are free, okay, so that's why here aldehyde and ketone group are free. Okay, okay, then what we can call, that will be called as a reducing sugar. But in case of sucrose, what happens? The first carbon of this pyranose ring and second carbon of this furanose ring, they are already been used up in forming glycosidic bond. So that's why what we can say, that this sucrose will be the non-reducing sugar. Now what is the importance of it? See, this concept is going to be very much important. See, what is the importance of reducing and non-reducing sugar? That we have a test, and that test is Benedict or Felling's test. Okay, so what happens? That Benedict or Felling's test, this is actually done for the detection of diabetes. Okay, so what is happening here, that in this Benedict's or Felling solution, what can we have? The cuprous ion. Okay, if the cuprous ion can be reduced to copper ion, then this type of sugars will be called as the reducing sugar. Okay, and only the sucrose, as the sucrose don't have the free aldehyde or ketone group, that's why what we can see, that sucrose is a non-reducing sugar. So those monosaccharides or disaccharides, they have a free aldehyde or ketone group, they can easily show precipitation in the Benedict solution. Precipitation means, suppose this is a test tube. Suppose we have the diabetes test. Okay, so in this diabetes test we have taken the urine sample. So this is the urine sample, and in the urine sample this individual have glucose. So this obviously glucose will be present in the urine if he is diabetic. So if you drop few of the Benedict solution into this urine sample, then cuprous ion will be present in the Benedict solution, and glucose present in the urine, it can reduce cuprous ion to copper ion, right? So whenever cuprous ion will be reduced to copper ion, some precipitation will be seen. So if precipitation occurs, that means that individual have glucose, and if glucose is present, that means that patient and the individual is diabetic. So that's why this test is very much important. You have to know the reducing and non-reducing sugar. Obviously, on all of the monosaccharide, they are not having glycosidic bond. As they don't have the glycosidic bond, that's why what we can see that all of the monosaccharide, remember, all monosaccharides, they are reducing sugar because they have the free aldehyde or ketone group, isn't that? So all of the monosaccharides, they are the reducing sugars like glucose, fructose, galactose, all of them, they are the reducing sugars. But on the disaccharide, if you see the disaccharide, we have seen the structure of three disaccharide. One is lactose, maltose and sucrose. From this you can see lactose and maltose, they are the reducing sugar. But what is the exception? Exception is sucrose here. Sucrose, as the aldehyde or ketone group is used up in forming the glycosidic bond, so that's why the sucrose is a non-reducing sugar. Okay, remember, this is going to be very much important. Okay, in the exam this question generally comes, what is reducing and what is non-reducing sugar. After that, the last part of the carbohydrate we will see is the polysaccharide. I already have written all these forms, but yes, we need to know what are the polysaccharides. Polysaccharides are the having more than 10 more monosaccharides. Okay, more monosaccharide units are attached to each other by glycosidic bond. Okay, it can be branched, it can be unbranched, it can be a homopolysaccharide, it can be heteropolysaccharide, but it is going to have more glycosidic bond. Okay, so in case of polysaccharide, they have two functions. Basically, they are found in the biosphere or in the animal body in two basic form. One is their function is storage. Okay, either it can have the function storage function or it can be structural. Okay, it can be either storage or structural type of polysaccharide. So these are the two basic function in case of polysaccharide. They are either storing something or they are helping or making the structure of this compound or that organism or that cell or that specific tissue. So these polysaccharides are basically divided into two groups. One is homopolysaccharide and another is the heteropolysaccharide. So what is homopolysaccharide? If the monosaccharide is only one type, suppose the whole long chain of this glycosidic chain we have, and in that glycosidic chain we have only one type of monosaccharide. Suppose only 30,000 glucose is present, which is present in the glycogen, that will be homopolysaccharide. But in this long chain, if different monosaccharides are forming the bonds and a long chain or a polysaccharide has been formed, then it will be called as heteropolysaccharide. So what is homopolysaccharide? It is going to have the same, okay, it is going to have the same monosaccharide. Okay, but if different monosaccharides are present, then it will be called as heteropolysaccharide. So first of all, what is homopolysaccharide? We can have only a same type of monosaccharide form the glycosidic bond. Okay, now some of the examples we will see. First of all, this is the example of a storage polysaccharide. What is this? Glycogen. So we know that this glycogen is a storage polysaccharide because it is the stored food of animals. All the stored food of animal is glycogen. So glycogen is actually formed by 30,000 glucose molecule attached to each other by glycosidic bond, right? And a very interesting thing we can see that it gives red coloration with iodine solution. So this is glycogen which is actually formed or gives characteristic red coloration with the iodine solution. Yes, we have the next type, that is starch. starch is going to form some helical structure. Okay, so they are forming some type of helix, so it gives blue coloration with iodine solution. So this is going to be very much important. Along with starch, you can see what is the function of starch. It is also storage food in the plants, isn't that? So one of the storage polysaccharide, another example will be starch. Glycogen is the stored food of animals, starch is the stored food of the plants. So it gives blue coloration with the iodine solution, and glycogen gives red coloration with iodine. So these two are very much important. One example we also have, that is cellulose. Okay, cellulose is going to be very much important because it is the most abundant carbohydrate in the whole biosphere. Okay, so what is cellulose now? The cellulose will be found in the cell wall of the plant. Okay, in the plant cell, if you see the plant cell, the outermost layer will be the cellulose. So cellulose will be the structural polysaccharide. Okay, so cellulose, as it is a unbranched structure, it is made up of 6,000 structures, monomers, that forms the unbranched chain. Then it will be a polysaccharide, that is called cellulose. Cellulose do not give any type of helix. Any helix or any branch structure will be not formed. Okay, so as it is not forming any branch structure, so that's why it is not possible it can form any type of coloration or give any coloration with iodine. So remember, this do not form, or they do not form any helix, so that's why it cannot give any coloration, right? Remember, as it is not having any helical structure, so that's why it cannot give any characteristic coloration with iodine solution. The next thing we need to know, this is another type of storage polysaccharide, that is inulin. Remember, inulin is a polymer of fructose. Okay, it is found by forming fructose that formed a glycosidic bond, and after that it formed the inulin. So it is actually the storage polysaccharide of the root. So storage polysaccharide, we have another example, that is inulin. Okay, so inulin is a storage polysaccharide which is found in the root. So it is not possible that our body absorb inulin. Our body cannot absorb inulin in any case, right? So that's why it is used for testing the kidney function. If in any case, suppose few of the solution having inulin is given to you, you will take it, and after that, this after suppose some time, after a unit period of time, suppose urine is been tested, then in that urine we are not going to have inulin. That means somewhere the kidney is not able to filter the inulin, because inulin can never be absorbed, so it has to come out with urine. But in urine, if it is not present, that means the kidney is not properly functioning. So that's why inulin is used to check the kidney function. Remember, this is very much important. Obviously it will be found in acid soluble pool or acid insoluble pool. This question came. It is found in obviously acid insoluble pool because it is a polysaccharide. Next one, chitin. Chitin is the second most abundant carbohydrate in the whole animal kingdom, in the whole biosphere. What is the reason? Because this chitin is found as an exoskeleton in different animals. Like in case of cockroaches, the exoskeleton is going to have chitin. A number of animals have chitin. Okay. Yes, this chitin is actually a nitrogen containing substance. It is also a storage polysaccharide. It is a structural polysaccharide. It gives the shape or the structure of the exoskeleton. So it is a structural polysaccharide. Example is chitin. So these are all the types of polysaccharide, homopolysaccharide we have seen. After that, let's just move to the heteropolysaccharide. So one example we have, that is peptidoglycan. Okay, peptidoglycan is actually formed by alternate amino and sugar molecule. Next one is hyaluronic acid. It is giving very much important for our body as it gives toughness and flexibility to cartilage and tendon. Next one, agar agar. Already all of us we have heard about it. It is a mucopolysaccharide which is obtained from red algae. So these are all the types of heteropolysaccharide, because it is formed by different type of monosaccharide. It can be a long chain of glycosidic bond having glucose, fructose, galactose, all of it, or more than one type of it. Not only glucose is forming the whole chain. So that's why we have two types of polysaccharide. One is the homopolysaccharide, second one is heteropolysaccharide. It can be divided into two types function wise. It is depending on the function, it can be either storage or structural. So next very important thing, two important thing I need to tell you, that the cotton fiber is going to contain around 90% of cellulose, where wool contain only 25 to 50% of the cellulose. So sometimes this question came actually, which among the following plants is going to have maximum or 90% is of the cellulose? It will be the cotton plant. Okay, so that's all about the carbohydrate. We have seen the types of carbohydrate, monosaccharide we have seen, oligosaccharide, the examples, disaccharide we have seen, and then the polysaccharide. So these are all about the types of polysaccharide we have discussed. That's all about it. Except this, no more questions will come. So that's all about the types of carbohydrates. So that's all about the carbohydrate. I hope you understand. Thank you.